Now we are turning to Nigel Dyer. He's a very familiar face. Okay. Nigel is from the United Kingdom, and he's one of the persons. He only missed one of the water conferences. So he should be very familiar to all of you, or to most of you. And he's talking about a very exotic protein, the Ecto-Nox protein. And he made a sort of secret out of it, because I wanted to ask him before what it is all about. But he told me, "Oh, me see," and I have to talk about it. So
you might want those.
Yes, I will sit here now. Enjoy his talk.
Thank you. As many other people do, I will thank Jerry for inviting me to give this presentation, and in fact to thank Jerry for organizing this whole series of conferences, because it has taken me on a very unexpected journey, because I turned up at the very first water conference not with any particular interest in water. I was there because I was associated with Vermont Photonics, who were the first sponsor of this series of conferences, and they invited me to give a talk at that conference, and I gave a talk about cell division, which at the time I thought had nothing at all to do with water. I now realize I was wrong, and it had a lot to do with water, but that's a talk for another time. But as I listened to the talks at that very first conference, I quickly realized, as we all realize, that there's something very interesting going on with water. And I wanted to be part of this club of people who were trying to put together this jigsaw, these different pieces of information about water, to work out what this jigsaw actually looked like. This is me at the second conference. After the first conference, I knew I had to come back, and I've been back to every conference but one. And there's a few familiar faces there in the picture. What I want to talk about is a couple who I was very privileged to meet at a couple of the conferences, and I want to talk about their work because I think they have a very important piece of this jigsaw which we need to remind ourselves about. Now, they are James and Dorothy Moraine, and I'm going to tell you a little bit about the experimental work that they did, which they explained in their talks. I'll talk a little bit about the hypothesis that they came up with to explain the results that they obtained. And then I'm going to explain why I think that hypothesis may be slightly wrong. And I'll talk a little bit about my ideas as to why it was that they got the results that they did. Now, like me, James and Dorothy did not expect to be going to a conference about water and to be talking about water, because that's not what they thought they were working with. They were working with Ecto-Nox, a protein. Now, we've heard about a number of proteins in this conference. We've heard from Jerry Pollack about myosin, the motor protein. We've heard about catalase from Lionel, which is the protein. It's an enzyme. So, like all enzymes, it's a factory. It takes some input molecules and it converts them into some output molecules, some different molecules. And the protein that James and Dorothy Moraine were working with is Ecto-Nox. Ecto-Nox is a very interesting protein in that it doesn't do just one job, it does two jobs. The first job is that, does this pointer work? Oh, a very small pointer, yes. The first job is that it breaks the sulfur bonds between some proteins. And the second job is it hydrolyzes NADPH. Now, for the purposes of this talk, we don't need to be concerned about the details of what those two processes are. They're just very different from each other. And it's very unusual for a protein to be doing two such radically different jobs. And the second strange thing about it is that it doesn't do these two jobs at the same time. It switches backwards and forwards between the two jobs. So it'll spend 12 minutes doing one job and then 12 minutes doing the other, switching backwards and forwards. And this is a graph from one of their papers showing that switching backwards and forwards between the two jobs. Now, so that means that the Ecto-Nox protein is acting like a clock with a 24-minute cycle, 12 minutes, 12 minutes. Now, in biology, if you have a clock, that usually means it's associated with a very complicated cycle where one thing happens which causes another, and so on. This is a representation of the pathway behind the circadian rhythm that gives us the 24-hour clock. But Dorothy and James quickly realized that their clock was different because it would work if you just had the Ecto-Nox protein sitting on the cell membrane and the water, the buffer solution within it. So it was just really the protein and water. And they were beginning to realize that the water was playing a very significant role in what was going on, which is why they then found themselves coming to a water conference to find out a bit more about water. Now, one of the experiments that they then did in order to try and understand what the relationship might be between water and the protein was to look at the bond length between the copper ion, which is at the center of the Ecto-Nox protein, and the water molecules that are around it. And when they did it, they came up with this the result of the experiment was this very complicated graph which shows a repeat every 24 minutes. So it did look as if this was somehow associated with this 24-minute clock. When I saw this graph, I thought, I've seen graphs like that before, because if you take two sine waves of different frequencies and you add them together, you get something like that has a beat frequency, which is at the frequency that is at the difference between the two frequencies. So if you go back to their complicated waveform and you superimpose a beat frequency type waveform, you actually get a very good match. It's not perfect, but I think it indicates that that's probably a hint as to what is going on there. And in fact, I was explaining this to James and Dorothy Moraine in that photograph you saw right at the beginning of the slideshow. That's what we were talking about at that time. And in fact, you can see that beat frequency effect in a number of the different experimental results that they present in the papers, because all of their work is published in papers in various journals and described in a lot of detail. For the next part of the talk, I need to introduce the idea of the two water spin isomers, because it was discovered in the 1930s, or they worked out in the 1930s, that there are actually two forms of water molecules. And this is actually picked up on one of the posters out in the poster session. You have ortho water molecules, which is where the spins of the protons in the two hydrogen atoms are pointing in the same direction. And you have the para water molecules, where the spins are pointing in the opposite direction. And in some water, such as you have here, you will have a mixture of 3 parts of ortho to 1 part of para. Now, normally you don't need to take you don't need to know about this, because in almost all respects, those two water molecules behave in exactly the same way. So it makes no difference to the chemistry and the physics or so on, by and large. So yes, here's some water with the mixture of 3:1 ortho to para. Now, just over 10 years ago, some papers emerged from groups who were working in Russia who claimed that it was possible to actually separate out those spin isomers within liquid water. So you could have a glass of ortho-enriched water and a glass of para-enriched water. And as part of that, they presented some work about this photon spectroscopy, which is a way of measuring the concentration of the ortho and para water within any particular sample. And the two were very much linked together. Now, James and Dorothy met some of these people at a conference, and they decided that maybe this was lying behind what was going on with the clock. One of the other things I should say is that when they separated the water into the ortho and para, they would have a half-life of about 15 minutes, and if you left it, it would go back to the standard 3:1 ortho-para ratio. And what James and Dorothy decided with these other guys from Russia must be going on is that the Ecto-Nox must be interacting with the water such that the Ecto-Nox was causing a change in the ortho-para ratio, and then that change in the ortho-para ratio was affecting the Ecto-Nox. So there's this cycle backwards and forwards would be at the heart of the clock that they were looking at. And that would actually solve another problem, because when they measure the characteristics of the Ecto-Nox protein, they're not just measuring one protein, they're measuring tens of millions of protein inside their sample. And for them to get the results that they're doing, all of those proteins must be ticking along synchronously. So you need to have something that synchronizes them all. And if they're all sitting in the same water, and the water is going, the changes in the ortho-para ratio are changing synchronously, then that would keep the proteins synchronous. So this is all was beginning to fit together. For photon spectroscopy is very difficult and complicated. So what James and Dorothy Moraine decided to do was use a very standard way of measuring the difference between the ortho and para molecules. And that is to use spectroscopy, because the ortho and para molecules spin at different frequencies. So they absorb light at different frequencies. So you can use spectroscopy to measure the relative concentrations of the ortho and para water. And they were going to do that in the water vapor that's immediately above the sample containing the Ecto-Nox protein. And they would look then for the variations in the ortho and para lines, which would tell them about the changes in the ortho-para ratio. So they did the experiment, and these are the results. They are messy. They're not as clear and as nice, I suspect, as they would have liked. They're not as clear and as nice as I would have liked. But nevertheless, they did some detailed analysis of it and did show that that same complicated beat frequency effect did appear to be there, which seemed to confirm their hypothesis that is somehow linked to changes in the ortho-para ratio. But I think there's a problem, because I've spoken to a number of people who know about ortho-para water and so on, and the idea that there can be a change in the ortho-para ratio within the water, within the liquid water, doesn't seem to be correct. That I'm fairly convinced of that now. And there are also problems, we think, with that particular technique of measuring the ortho-para ratio. So, where does that leave James and Dorothy's results? Well, the technique that they used for doing the measurements of the ortho-para ratio, that's okay, that's kosher, that's a standard technique. So it does look as if the results that they got are probably okay. Now, if you look carefully at the results, and this is taken from one of their papers, what I think you can see is that the complex waveform is actually just in the ortho measurement, and that the para measurement is largely experimental noise. So I think that what they were measuring is something that just was picked up by the ortho molecules. Actually, if you go to the paper and look at their graph, they say that it's the paramolecules, but they've labeled the frequencies the wrong way round, which you will find if you go back to the original paper that they quote for the information. So it's actually, I think, the ortho molecules that are picking up some kind of signal. So I think now there's a new way, possibly, of interpreting the results. And that is that there is something going on within the sample, within the Ecto-Nox and the water, that is then being picked up by the ortho water molecules in the water vapor above the sample, but not by the para water molecules. And that is, I think, the reason why they got the results that they did. So if that's happening, what are these signals that the ortho water molecules are picking up and not the para? Well, I wonder whether there may be some kind of macroscopic vibrational mode or coherent oscillation mode within the water, and that is what the water molecules in the water vapor are picking up. Or rather, there's not a single oscillatory mode, but there are two oscillatory modes at right angles to each other. Because they're at right angles to each other, they would be independent of each other, and they can be at two different frequencies, perhaps at two different frequencies that generate that beat frequency effect. And the Ecto-Nox protein, the copper, is then picking up the sum of the two frequencies, which is why you then get that beat frequency effect, and that is then driving the 24-minute clock within the Ecto-Nox protein. So all of this would then suggest, perhaps, that there is this resonant mode that involves, that is taking place within the water, that is being picked up by the ortho water molecules in the water vapor above the water, and not the para. So what would the implications be of this? This would suggest, well, if this is taking place, then the fact that the ortho and para molecule difference is key to this would suggest that the spin of the protons is key to understanding what's going on. And this would suggest that we're talking about some deep quantum mechanical interaction that is going on that is involving spin. And maybe the Ecto-Nox results that James and Dorothy have come up with have shown that there is this long-range quantum mechanical interaction taking place in water that involves the spin of the hydrogen atoms. And maybe that, in part, might explain some of the other subtle and interesting effects that we hear so much about at the water conference, such as perhaps being part of the explanation for the exclusion zone, or the coherent domains, or nanoassociates, nor a component of all of these. If there are no such interactions, if what I'm saying is complete rubbish, then we have to go back to square one and look again for what is the explanation for the 24-minute Ecto-Nox clock that clearly involves the water in some way. What is the explanation for the beat frequency effect that we see in the results? And what is the mechanism that causes the clocks for all of the different Ecto-Nox proteins to be synchronized? But one thing that I am confident in is that whatever that explanation is, I don't think it involves a variation in the ortho-para ratio of the water. Of course, my acknowledgements, I have to start with James and Dorothy Moraine for all of the wonderful work that they did over many years. And in a sense, this is in memory of James, because sadly he died in 2016. And of course, acknowledgement to Jerry for organizing these wonderful conferences and for inviting me to speak. Thank you.
So thank you very much.
It's a guy not here. He's coming.
Thank you very much for your presentation.
He'll check that it's turned on.
So, and thank you for being in time. This was great. So we have a lot of time for questions.
Clocks, you see, clocks.
What questions do we have? Is there somebody? Yes.
Thank you, Nigel, for brushing up on this particular topic on the Ecto-Nox protein, because we talked about it quite often. We have, yeah. I wonder, because having done this kind of brush up again, I wonder if you have a coupling, a resonant recognition coupling between the ortho frequency band, because you said there's a shift in the absorption bands between ortho and para, and the Ecto-Nox resonance mode. And I think these coupling probably are crucial in understanding why these chronobiological cycling is taking place.
I think you're absolutely right. But in order to start investigating that, we have to sort of come to an understanding of what it is that we are investigating. So to a certain extent with this talk, I'm just trying to establish the first step in that process and to say, I think there are some reasons why when we start looking at that, we need to start thinking about the spins of the hydrogen atoms and interactions that involve those and how we might measure them, and then move on to those sorts of
because this brings me automatically to the second part of my question, which is that in that case, you probably have coherent domains coupling.
Yes.
And the water body itself is just acting as a transducer.
Yes.
And an amplifier, so to speak.
I totally agree, yes, yes. We've sort of discussed that, so yes.
Further questions? You.
Thank you very much, Nigel. Very interesting presentation. I would like just, probably not answer, but a small comment. It is very nice that you included these easy water coherent domains and nano-associates, and it seems to me that they allow to connect with ortho-para this problem, because if we have coherence, then should be some fractionation. For example, in coherent domains, I don't think there is a mixture of ortho-para. Some should be there, and probably those who are magnetically susceptible. And probably just to see if ortho-para really and their feeling of environmental magnetic fields is just to use Konovalov methods of shielding from from environmental magnetic fields, and to see if this will violate this period of 24 minutes.
Yes, I think the magnetic field interactions that Konovalov and others show is very important, because I think that'll be part of the process of inducing some kind of symmetry breaking, so that, for example, the spins may become slightly more aligned in a particular direction, but it takes something external, like the magnetic field, to induce that symmetry breaking. And that's why the magnetic fields provide, I think, a very important clue as to what is going on.
And also, I would like to remind that Persson, if I'm not mistaken, he has demonstrated that there is fractionation between ortho and para when you have easy water. In easy water, in water of hydration, one of these forms of water, I don't remember which one, is in great excess over another.
That's the point where
it is one is rotating and another is not.
Yes, I'm less confident with those results now, but I think it may be that if we go back and look in detail at the measurements that were done, we may want, like I have done with the Moraine's results, to sort of reinterpret and reassess those results, because I think it might be that it's telling us something slightly different from what we thought it was telling us, but that the ortho and para difference is still key to understanding what is going on. That I very much agree with and was trying to present here.
Thank you. Further questions? Yeah.
Thank you, Nigel, for that wonderful presentation. First of all, this is a comment because in simoscopic imagery, I very often see with specific sound frequencies injected into water a kind of a scintillating effect in the background, and I have never known what this scintillating effect has related to. I've discussed with many people about the possibilities of atomic physics and resonance and so on, but the big problem has always been that the frequencies that we're talking about in atomic physics are so high in relation to sonic frequencies, there seems to be no possibility of resonance. But bringing up what you have just described here seems to provide a possibility for why we are seeing this scintillation. What is the sort of period of that beat frequency? What are we talking about, Nigel?
Here, the beat frequency that we're talking about that the Ecto-Nox is using is the 24-minute. But the audio frequency type effects that you were talking about was actually one of my very first clues that set me looking in this direction. Because if you start looking at the spin of the hydrogen atom, then that precesses in the Earth's magnetic field at a frequency of about 1 kilohertz. In other words, right in the middle of the audio frequency band. And we have so many experimental results that seem to suggest that things that are working in the audio frequency band are somehow able to affect atomic physics. So, you know, how can that happen? But that might be how we can link the two together, that the precession frequencies in the Earth's magnetic field are in that sort of range.
How interesting. Thank you so much for that, Nigel.
Further questions?
I don't know where the 24 minutes comes from.
There is another one.
I made some calculation for the range of your research, and my question is, what is the connection of your research and biophoton research of Professor Fritz Albert Popp?
I don't know, but I think there is a connection. I'm fairly certain there is. I went to Neuss for a couple of the biophoton conferences and learnt so much there, and like a number of people like Vladimir were convinced that there is a strong relationship between the two. And certainly in what I'm moving on to look at, that is something I will be very much looking at, but I don't have an answer to it now.
There's another question.
I have a very short comment. I was thinking about the question about sound waves. Perhaps, but I'm not sure, just an extemporary thinking. Perhaps the para water, which is less sensible to orientation, because spins are up and down, so perhaps those molecules are more free to react to sound waves. So if frequency should be, matched, maybe it's the para water, which is also maybe the reason why it is like a background in these graphs you have shown. Because the other one, there is, for the ortho, a magnetic moment associated with the orientation of spins. In the other case, the magnetic moment would be zero of that special spins, hydrogen ones. So I don't know, but thank you for your question.
That is a very interesting idea and worth pursuing. To a certain extent, my objective here was just to introduce this idea so that people can then take it away and see whether it might actually help in the particular area that they are working with. We're saying, yes, because if that is the case, as you have just done, then maybe that might be another way of thinking about what's going on in my particular context, my particular experiment. Yes.
Yeah. Thank you.
I think we go on in order not to get late. We dived very deeply into physics with the presentation of Nigel. Thank you very much again. The next speaker is